Hi everyone, my name is Abdul Malik Setiawan. I'm a PhD student in Anatomy Department of UKM, Malaysia. Today, I would like to talk about my study, OCT4 and BMP4 expressions in human adipose-derived stem cells isolated from subcutaneous and visceral fat. Corneal blindness is a condition when there is an opacity in the corneal epithelial layer caused by trauma, infection, and autoimmune disease. The gold standard therapy for corneal blindness is corneal graft or limbal stem cells transplantation. However, because of the shortage of donor, researchers try to find alternative. One of them is by stem cell therapy. Adipose-derived stem cells, or ADAC, is one of the stem cells that is abundant and easy to isolate in human body. A recent study showed that optimal binding transcription factor, or OCT4, is regulating the pluripotency of most pluripotent cells. One of them is in ADAC. Other studies showed that bone morphogenetic protein 4, or BMP4, is essential for early eye development, and it's also can be used to differentiate many pluripotent cells towards to ectodermal lineage. Therefore, the study aimed to investigate the expression of OCT4 and BMP4 in different fat locations, which is subcutaneous and visceral. There were 6 females that agreed to participate in this study that were undergo a cesarean section in ONG department in Hospital Chancellor Tuanku Mukhris, UKM, Malaysia. The surgeon collected the subcutaneous and visceral fat, and we digested the fat using 0.3% collagenase type 1 for 4 hours at 37 degrees Celsius. Then we inactivate the enzyme using culture media. We wash the PBS and filter with 80 micrometer cell strainer. The cell pellet is known as the SVF, the one that containing the ADAC. Then the SVF is suspended in 1 ml culture media, consists of DMF12, 10% FPS, 1% vitamin C, 1% antibiotic antimitotic, and 1% glutamac. And then the cell seeded into 6-well plate and cultured in incubate in 37 degrees Celsius at 5% CO2 until passage 4. When the cells 90% confluent, we isolated the RNA using triazole methods, and then we synthesized the cDNA. Then we use the qPCR analysis to measure the expression of OCT4, BMP4, and GAPDH gene as a reference gene, and we use the ADAC cell line as a control. We then calculate the relative expression of the those gene in subcutaneous and visceral compared to the cell control using the delta-delta-CK methods. And then we use student test to evaluate whether the expression between the visceral and the subcutaneous is different enough or significant enough. From our samples, the average age were 35 years old, and then the average height was 158 centimeters, and the average weight was 71 kilogram. Based on the relative expressions, the OCT4 expression of ADAC and subcutaneous and visceral ADAC is higher compared to the cell line to the control. And if we see the relative expression of the visceral ADAC shows higher compared to the subcutaneous in 4 patients, but only 2 patients that's statistically significant higher expression of OCT4 compared to the subcutaneous. The relative expression, the relative BMP4 expression of subcutaneous and visceral ADAC compared to the control is higher. And if we measure the relative expression of visceral ADAC compared to the subcutaneous, most of them is higher in visceral ADAC compared to the subcutaneous. 4 patients shows significance, higher expression of BMP4 compared to the subcutaneous ADAC. So we know that the OCT4 is a marker of stemness, and Kobayashi in 2020 2020 showed in his model that BMP4 could use to initiate the differentiation of iPS cells towards the ocular surface epithelium. And this differentiation might from activation of TGF-beta signaling pathway. So these findings is in line with our finding that visceral and subcutaneous ADAC express higher or maybe similar level of OCT4 and BMP4 compared to the control, which is ADAC cell line. But the OCT4 expression in visceral ADAC is higher compared to the subcutaneous. And we also know from Ritter 2019 that visceral ADAC compared to subcutaneous ADAC express higher stemness associated genes and also has higher activation of sonic hedgehog pathway. And the sonic hedgehog pathway signaling is known to promote limbal stem cells and corneal epithelial cells proliferations. This is in line with our finding as well, that visceral ADAC express higher OCT4 and BMP4, even though the stemness gene is different, but it's in line. And if we compare to the subcutaneous, the expression of OCT4 and BMP4 is higher in visceral ADAC. Thus, we can assume that visceral ADAC has higher stemness and potential difference in the capacity if we want to differentiate into corneal epithelial cells. In conclusion, our study, the visceral ADAC have higher stemness characteristics and ectodermal differentiation capacity. So therefore, it could be a better choice if we want to use the visceral fat as a source of MSC, if we want to differentiate into another cells, especially in corneal differentiations. So I want to acknowledge the Citrum for ADAC cell line procurement, ONG Department of ACTM for sample collection, and Anatomy and Physiology Department staff and fellow students for their help for this study happen. This is my reference. Thank you. I would like to answer all the questions from the audience. Thank you.